sort of semi-retired now. I'm sort of a piece of history. No, not that great. But it's actually a great joy to be here with you this morning to talk about an issue. Basically about the genetics of intelligence. It's not something you hear a lot about these days, but when I was a kid, and I'm talking about the late 60s when I first got into this business, doing my Ph.D. It served two purposes. One is, it was probably in quotes the most important thing about behaviour that anybody was really interested in and cared about. So, one. I mean, it's so fundamental that the ability to solve problems, deduce correlations, and so on and so forth. As Peter Spearman said back in the to see relations and deduce correlations, how he defined intelligence back in the early 20th century. It's so fundamental to humans, and probably is what sets us apart from the rest of the animal kingdom. If I may be so anthropocentric boast for a moment. And so, if you wanted to address the fundamentals of what made humans human, the ability to think, reason, and everything else seemed to be something you really needed to know about. And there was, as probably almost a century of psychological research shown, whenever anybody tried to measure the construct of intelligence, there was enormous diversity. And that was just a sort of fact of life and nature. So, as people began to think about the implications of biology for humans, it was a natural place to begin digging. In fact, the history of the subject goes back to about 1865. Francis Galton, who was, like me, a Brit. He was a rich Brit, and so he didn't have to work for a living, and so he could sort of spend all his time thinking and having fun with science. Wrote a masterpiece of work, in so many ways a masterpiece of work, called Hereditary Genius. And this was in 1865, and what he did was to collect the pedigrees of people who, by the definitions of Victorian England, met the criteria of genius. They were people of exceptional accomplishment. Now, of course, because it was Victorian, that was, only men qualified. So it's there's an element of inbuilt sexism in this. But so he identified people who were great mathematicians, great judges, even great clergy, God help us, and including people who succeeded in wrestling contests and that kind of thing. There's a whole collection of pedigrees of North Country wrestlers. And what he did was to trace the family histories of these people and show that if you defined genius in almost any kind of way, when you looked at the family histories and also the descendants of these people, the prevalence of genius amongst the relatives of genii was so much incredibly inflated over the risk of being a genius in the population as a whole. If you think of genius as a disease, then you sort of say, well, okay, 1% of the population might qualify as a genius. Then, you were looking at risks of like 20%, 10%, depending on the degree of relationship. So, began to build this picture of it running in families. And of course, this was the era of Darwin and Mendel, so naturally the assumption was, and Galton was quite self-critical about this, the assumption was it ran in the families, it was genetic. But of course we all know that's not really true, and Galton knew that wasn't really true, because he thought, well, people who share their genes probably also share cultural advantages or disadvantages, and so you have to take into account the non-biological transmission of information down pedigrees. And in fact that's what led Galton also to invent the twin method, which we will talk about a lot today. But really the reason I wanted to talk about the measurement, the genetics of intelligence today is twofold. One is because it's actually what people are most excited about at one of the most transformative times in our subject. In the late 60s and extending beyond that through the 70s and maybe even into the 80s, there was enormous aggravation around the idea that behaviour might even be genetic. and particularly something as intelligent. I mean, we all want to believe in the infinite plasticity and modifiability of humans. You know, and we want everybody to be able to sort of, you know, be as good as you can be, kind of thing. And so the idea that, you know, people might be operating under either biological or social, biological constraints or biological, you know, disadvantages was, you know, actually considered inherently, basically fascist. And indeed, there were people who sort of, you know, kind of wanted to capitalise on that people are biologically stupid and therefore we don't need to do anything about them. And in fact, you see elements of that in some of the political field today. But I mean, believe me, when I was a graduate student, we're now talking, what, 1968 through 1970, and there was a postdoc into the sort of 70s. People were being abused, I mean, physically. I mean, I remember Hans Eysenck, we'll say a little bit about, in fact, the author of this book in which these chapters occur. Hans Eysenck, I remember coming to give a public lecture in Birmingham University, where I was, and I mean, you know, people rushed up and spat at him. You know, and there were signs that were written, sort of great whitewashed signs on the library: "Fascist Eysenck, get out of Birmingham." Now, the irony, of course, was that Eysenck was Jewish by birth. He was German by birth. He actually left Nazi Germany, you know, because of all that was going on there. And in fact, one of the reasons he left was because when Hitler was starting to promote anti-Semitism, he wrote a little essay, I think, in school for his because I think his teacher had sort of made some anti-Semitic remarks about, you know, Jews are not, you know, cowards, that kind of stuff. And so what Eysenck did was to count and research the number of Jews who won Iron Crosses in World War I, and show that in fact they were as brave in terms of winning Iron Crosses in Germany, or maybe even braver than the average German, you know. And so that didn't go down very well with the Nazis any more than, you know, Jesse Owens, I think he won the Olympic track gold in Berlin when Hitler was sitting in the stand. So, you know, there's a lot of aggravation about that, particularly, of course, in the States. But there was a lot of that in England too. And I came into this subject at that point, in about 1968. I first met behaviour genetics as an undergraduate in my final year as a geneticist in Birmingham. And that actually really leads me to talk a little bit about the cast of characters. Because firstly, it was an issue of enormous social and, you know, for me at least, less than social, but actually just philosophical importance. So you want to understand why I got into this game is because you want to understand what humans are about. You know, you've ignored the DNA at your peril. Now, I mean, DNA had been around that long already when I was a kid, you know, but you couldn't ignore genes. So I thought, or at least, you know, genetics were going to change the way we thought about the world. You know, and if you care about thinking about humans, you've got to take genetics seriously, which is sort of, you know, where I got into this. And this was a kind of model system, you know, which seemed to have all those implications. You know, if you figure this one out, you're going to say something fairly serious. Probably in parallel, though with slightly more complications because of the nature of the phenotype, you would probably identify in that period schizophrenia as being an issue where there was a comparable sort of story, and in a sense just as much aggravation. You know, I mean, in the 60s and 70s, Ken Kenner's much more the expert on that history of sort of psychiatric than I, but I think he would say that there was an equal period of transformation when people went from a predominantly social paradigm for the etiology of schizophrenia to a genetic paradigm. And there was somewhat less, perhaps, of the aggravation, but nevertheless, you know, it was there. But for intelligence, it was a major issue. Big deal, you know, classic phenotype. The other reason was that in that period was a period of enormous intellectual ferment about actually how you do behaviour genetics. And I want to spend a few minutes just showing, for example, that pretty much all the characters that you hear about, in fact you actually learn from, you know, Mike Neal, me, Judy, Ken, Hermine, all these characters share a common connection which really first grew in this period, in that period, stemmed from the work that was being done in Birmingham, England, in the late 60s, early 70s. And the focus of our conversation, when we actually start talking about the data, is this chapter by, now unfortunately the late David Fulker. David died probably about 10 years ago with pancreatic cancer. But David was one of the sort of seminal characters in this film. We're going to look a bit at how all these things join up. But David was A, astonishingly smart, B, didn't give a shit about what people thought of him. So, you know, I mean, he would actually tell it like, sorry. You know, David, you know, really would tell it, God, I hate to say this word now, we've heard it so much in the nauseating political context, but David told it like it is, I mean, you know, in the academic community, and wasn't afraid to speak the truth to power, and professors and all those other people. So he's astonishingly bright. Also, he was a phenomenal teacher. And in a sense, the chapters in this book, we're only going to really look seriously at one of the chapters in this book he wrote, but are a didactic masterpiece. And the reason I'm using him is not because you know, I can't teach the subject, but because he teaches it so well. And they're beautifully written, and it's a great sort of example of scientific writing for people who are actually trying to teach a subject. So let's look just a little bit at what was going on in the culture of behaviour genetics in this period, and you know, just spend a little time talking about the story. And really, I guess there are two characters who probably, to whom all of us here owe our sort of origins, and they themselves were not really behaviour geneticists, but they were people who spawned and either directly or indirectly fostered the growth of behaviour genetics, and probably, I think it's true to say, without whom none of the people here, and none of the people who've gone out from here, would have survived. One is Kenneth Mather, who was a geneticist. Mather really, I think, probably thought all psychologists were stupid, probably thought I was stupid, because he was astonishingly bright. He was also a cantankerous old soul, and so, you know, and he wasn't, well, okay, I'll tell you one Mather story, okay. I had a graduate student, Christina Last, who had been working on genetic on social attitudes. And she gave a seminar to the department. Mather, by this time, had retired. He was going to be Emeritus, and he was a really right-wing conservative, you can probably imagine. And Christina gave this talk on genetics of social attitudes, and Mather, sitting at the back of the room, sort of said, you know, what do these things measure, you see? So I, springing to the defence of my graduate student, said, well, you know, what do the hairs on a fruit fly measure? Because Mather's spent all his life working on the number of hairs on a fruit fly, you see, the model system in genetics. And so you can see everybody in the department starting to duck behind the tables, a bit like when a guy comes into a bar, you know, with a pair of guns blazing, you know. I thought, oh my God, then you could see Mather sort of smoke like on everything. He said, it's got something to do with getting some useful work done, that's what. But he was actually one of the great characters in the subject. And so it's on the genetic side. Then on the psychology side, in London, this was in Birmingham, which we in England call Birmingham, not Birmingham, but Brum. Right, yeah. Because in fact it was originally called Bromwich back in the Middle. I know you guys don't go back to the Middle Ages, we do. Back in the Middle Ages, called Bromwich, and so Brummagem, and so we talked about Brummies. I'm a Brummie, okay, and Brum is short for Birmingham, okay. So Brum, and then Isaac worked in London at the Institute of Psychiatry, and he was a. You don't hear much about him now, but he actually, at the time, was a seminal thinker in the biology of behaviour, and he wrote significant works, for example, on the structure and measurement of personality. He did one of the very first, actually, interesting, one of the very first multivariate twin studies, looking at personality in twins and sort of showing actually that, all different measures of neuroticism seem to share a common genetic basis. And actually that's really prophetic, because now we think about, for example, of the common genetic basis of anxiety and depression. It's kind of prophetic early work. And Isaac had a student called Broadhurst. I'm not sure if it's a student or a colleague, Broadhurst. Peter Broadhurst, who retired rather young. And Mather had a student colleague called John Jinks. Now, John Jinks really is the sort of intellectual powerhouse who helped transform behaviour genetics from a cast of characters farting around looking at numbers to people who really thought about what this means genetically and how you go about constructing models for the influence of genes and environment on human traits, and particularly how you did the statistics. Because John Jinks was what was called, along with Mather, was a biometrical geneticist. Now Jinks actually was really interested not in humans, he was interested in genetics. And so he worked really on model systems. I mean, humans are a mess to work with, because they have that sort of, they mate with who the hell they choose. You can't inbreed them. You can't produce inbred lines. You can't conduct crosses, at least not legally. You have to take what you get. You can't randomise the environment. So humans are a mess, but you can do some beautiful things with plants, fruit flies, and fungi. And he and his colleagues and students in Birmingham really wrote the book on how you develop mathematical models and test mathematical models for the complex roles of genes on complex traits, like the number of hairs under the armpit of a fruit fly, or the amount of penicillin a fungus spews out into the medium, or how tall a plant is at the end of the season, or what time the first flower opens. Those are the kind, and because you could really manipulate these organisms, you could do developmental studies, and so on and so forth. So really, this guy, who was, when I first met him, had just become chair of genetics in Birmingham, and he, I think, very shortly after I met him, I think the age of 40, which is very young, got elected as a Fellow of the Royal Society, which puts him up there with Newton and Galton, and I mean the people who really are sort of the top of their field. I mean, these are not just smart people; they are the sort of genii who are recognised across the board for their. So John Jinks, FRS, also he was Dean of the Faculty of Science and Chairman of the English Science Research Council, which means that there's a head. As he once said, he was actually on the whole a very modest guy, but very smart. Wouldn't put up with anything from anybody. Once said, "I am the most powerful biologist in Britain." And it's true. But he's also probably the brightest. Now, so Broadhurst had done some experiments on. I think it was rats. Actually, it was Maudsley. Yeah, the Maudsley Hospital was doing some breeding experiments on rats, and so he got data which was sort of vaguely genetic and. What does he do with it? And somebody, in a fit of brilliance, said, well, actually, he said, you need to talk to this guy Jinks in Birmingham. And so there was a kind of a spiritual marriage where these two guys got together and started to say, okay, so what does all this genetics that that these people in Birmingham were working on, these beautiful ways of thinking through mathematical modelling in genetics and so on, what does this do for our understanding of behaviour? In this case, using rats as a model system. So, Broadhurst ends up as a chair of psychology in Birmingham. And Broadhurst had this graduate student, and we're now looking 19 this would be about 1965-ish, say. 1965-ish, maybe 1966-ish. Broadhurst had this student, David Fulker, who was born in London, came to work in Birmingham, and was really the first person to begin work under really Jinks, although formerly he was a Broadhurst student, really began to ask, how do we get biometrical genetics, mathematical, statistical genetics, for polygenes, lots of genes working at the same time, how do we get biometrical genetics to work for behaviour? And David had a remarkable sort of track record. I don't know anybody else who really shares in quite that way. He worked on fruit flies. He published a paper in Science on mating speed in Drosophila, about 1966-ish. He did his major work for his PhD was on the rat, doing what's called a diallel cross, where you take lots of different inbred lines and cross them together and do a lot of really cool genetics. And he did things like mashing up brains and extracting chemicals from the brain. I mean, he did a lot of really, I mean, in a sense, he actually did a bit of even neuroscience in genetics back in those days on rats. But also I think that we particularly recognise him as, particularly under the influence of John Jinks, did some of the first applications of the way biometrical genetics is applied, applying it to humans and human behaviour. And really, there's not a person, even if they don't know it, there's not a person in this, how many square feet you've got here, who does not owe their ability to look at data in the way we do around here, does not owe it to a paper which Jinks and Fulker published together in 1970. It was published in the journal Psychological Bulletin, and it's got a slightly odd title which represents where it was in history. It's called A Comparison of the Classical, and then MAVA, spelled capital M, capital A, capital V, capital A, and biometrical approaches to the analysis of human behaviour. And what that paper was about was took a whole range of data from human studies that people had tried to do genetics and behaviour and say, look, you're all farting around because you're not thinking about what the genes do. We have a theory, a mathematical model, which actually sort of goes back to Fisher in 1918, which predicts how genes will affect the similarity between different degrees of relationship. It's a simple model in a way. You know, it reduces a complex system to a few really simple constructs. And we have the statistical methods to test that model. And if it don't work, we have ideas about how to fix it and change it and modify it. And so really, what they did was to introduce into our thinking and methods the whole approach of model fitting, where you think about how you think the world might be organised, you develop a mathematical model, you collect the data and you say, what values of the numbers for the various effects emerge from applying this model from data? But not just that, but does the model actually explain the data? Now, I mean, any old fool can look at data and go, oh, I think it's that and that and that and a bit of that. I mean, that's what they're doing. But it takes a few more IQ points to say, okay, but how do I know whether I'm right or wrong? And after all, science is about the falsification of crappy models and the development of better ones. So really, this was a seminal piece of work. And into this mix, in 1968, came this young guy called Eaves. And this wasn't a major custom in the tradition, but I'm just putting my piece of the story in here, okay. Believe it or not, I just left seminary. So I'd literally just been ordained in the Church of England, but as an undergraduate, I'd already seen some of this work going on. I thought, gosh, this looks really cool. You know, if you want to talk about humans, you better damn well get into this stuff. And the opportunity came up. So I started my PhD on the Monday after, in 1968. And I really sort of got it from these guys. And I became the guy who, unlike David, of course, spread across the map doing humans, fruit flies, and I was the one who was charged with trying to make extend the human stuff. I mean, it was all right. But the point was, what are you going to look at? Well, later on I got interested in all sorts of other personality stuff, but at that point, IQ was the thing to look at. Now, just to fill in the story, okay, there's a whole bunch of other characters. Yeah, it's fascinating, because if you look around now, there's a guy called John Hewitt, who is now the director of the Institute of Behavioral Genetics in Colorado, and I'd say is our actually chief competition. Well, this is another piece of competition. There are a few more, but in the States, in terms of graduate students, Colorado is probably one of the places that gets, picks up some of the best. It's directed by John Hewitt. John Hewitt, believe it or not, was an undergraduate psychologist in Birmingham. Who got interested in genetics and so came to do his master's in genetics, went to London for his Ph.D. with David Fulker. Okay, so you get the drift of the way things are unfolding. Now, who else is on the scene? Well, you may or may not come across a guy called Andrew Heath. Andrew Heath is also a Brit. He did his PhD. He was sent by he was an undergraduate in Oxford who was sent by his supervisor, the undergraduate supervisor, Geoffrey Gray, to spend a few weeks with me in Birmingham, because he was interested in genetics of behaviour. And so I had a bit of an influence on Andrew. And then when I moved to Oxford, he became my graduate student. And to cut a long story short, he then came here, okay, so Hewitt came here for a spell as a postdoc and a young faculty member. Andrew Heath came here as a postdoc. Heath also was an advisor to a psychology graduate student, clinical psychologist called Silberg. And he was on her committee and sort of, pushed her in the right direction, or at least sort of fed the genes, I guess, which were already crying out and thirsting for genetics, right? Andrew Heath emigrated, from England, as did John Hewitt. Andrew Heath is now a distinguished professor of genetics in psychiatry at Washington University, St. Louis, probably one of the, another rather good place, and heads their program, particularly in alcoholism research, the genetics of alcoholism. I mean, off the scale, bright, scares the proverbial out of me, this guy, really. Right, and I think it pretty much scares you too, right?
Absolutely.
Yeah, absolutely. I mean, he's got a mind like a razor. Andrew Heath, who else is in there? Oh yeah, Mike Neal, okay, Neal. Mike Neal did his PhD with David Fulker in London. And then came over here, right? There was a guy called, my first graduate was a guy called Nick Martin. Nick Martin's an Australian, came to Birmingham to figure out how to do all his biometrical genetics, mate. G'day. And then for a spell he came here as a faculty member, and then went back to Australia after having married one of our graduate students. Georgia is now a pretty leading cancer geneticist in Australia. Nick went back to Australia, now heads up the genetic epidemiology program in Brisbane, Australia. And it's probably actually one of the most generous, and actually he introduced us, I think, didn't he, really? Mm-hmm. Yeah, right. And there's one more person, oh yeah, Hermine Mast. Well, by this time, this cast of characters, Martin, Heath, Eaves, Fulker, Hewitt and so on, and Mike Neal, were all realised they were onto something really kind of cool. People needed to hear about it. And so it was actually Nick's idea, Nick Martin, that we should organise a series of workshops. And actually a couple of guys in Belgium persuaded NATO to shake the trees and give us enough money to collect a group of interested students together in a place called Lindon in Belgium to teach basically statistical genetics the way we went about it. And in Lindon in Belgium there's this lovely young woman called Hermine Maas, who at that point was a graduate student, met Mike, who then became sort of, an item, and so, she came here as a postdoc, and all the rest is history. So I just paint that out there. Basically, pretty much all these people left England because, this was the land of opportunity. But these are all the characters in there. So in a sense, you see that there are lots of connections. We all owe this now. But there's a person really, I think we all owe a major tribute to is David. And the genetics of intelligence. And what we're going to look at, his work on genetics was sort of seminal to us. So just that puts this in context, as it were. And it's important to realise that all of us work within an intellectual tradition. Yeah, we've all done our bit, but we didn't invent the subject. And so it's really kind of important to recognise that, because actually buried in here there are some really smart people with some really good ideas. And in fact, going back before this, there were some really smart people having some really good ideas. And none of us would have existed without them. Now read on, the genetics of intelligence. And how do we measure it? Well, I'm not going to talk too much about the measurement of intelligence, but you've all done some kind of IQ test, right? You know what you do. Typically, it consists of a graded series of problems. And what you find is, if you give someone a series of problems of graded difficulty, firstly they reach a kind of stopping point where on the whole they start to get things wrong. And I put wrong in quotes because there's no right or wrong answers in this world, are there? No, right. Just good and bad guesses. And what you find is that typically, the same person on multiple occasions will tend to start to get things wrong at about the same level of difficulty. If you define difficulty as how many people get the question right. I mean, for example, you present a verbal analogy. Hand is to wrist as foot is to ankle. Yeah, ankle, right, yeah. Well, okay, that puts you in the top 50% of the population. Yeah, cool, yeah, right. But you get the idea, right? And so first it's pretty repeatable. People are pretty stable, on repeated occasions. The second thing, but not perfect. The second thing is that there's enormous diversity. Some people really keep going. I mean, they can get sort of problems that only sort of 0.5% of the population get right, and some people struggle with questions that only sort of 90% get right. And so what you have is a ranking. And what people really discovered was that actually, when you rank people in this way, you can assign, when you know how common or rare a score is, you can begin to say, well, rare means either they get a very unusual, extreme collection of good stuff, or a very extreme collection of bad stuff. Most of us in the middle, get a mix of good and bad, and we're the most common class. And so this led to the idea that perhaps underlying the ability to do the test, there was a normal distribution, continuous distribution of liability, if you like, to be either high or low on the scale of intelligence. And there was enormous diversity. And so it was reliable, repeatable, and in some sense pointed to underlying your ability to do, and I use the word well, without trying to be pejorative, on a test, may lie a trait that's not unlike height, continuously variable, really, and that could map where you were on the scale of this intellectual stature onto how well you do on the test. So, when people you take an intelligence test, you're given a so-called IQ, right? Now, IQ is scaled to be kind of like height, normally distributed, and it has a mean of 100, artificial, right? It has a standard deviation of about 15, artificial, right? But, you know, it's a way of thinking about individual differences in behaviour. So, you know, 2 standard deviations above the mean is 130. So, you know, it's designed so that roughly someone who can answer questions that only the top 2.5% of the population can do gets an IQ of 130. You know, someone who can answer those questions that only probably 0.5% of the population can do gets an IQ of 145, and so on. You know, 50% of the population will get IQ scores of 100 or bigger. So you have to think. You know, so in a sense, in psychology at the time, it was also one of the few constructs that you could actually measure that was A, about the human sort of problem solving, and B, actually had enormous, you know, sort of produced a reliable, repeatable measure, and so it became worth looking at for that reason. Aside from the fact that A, you know, it seemed to be problem solving seemed to be important. So the question is, how do you do the genetics? Well, you could, of course, start out, as probably people have done, you know, collecting data on relatives, you know, parents and offspring and that kind of stuff. But really, the most instructive first shots were attempts to look at twins. And if we look at David's chapter, I think it's table 1 in the chapter. Yeah. We see one of the first fumbling attempts to do behaviour genetics. It's actually table 5.1. It's the first table in the chapter. Okay. Chapter 5, table 5.1. One of the first fumbling attempts to do the genetics of intelligence goes back to this study of Herman and Hoffman. There were several early studies, but this is, you know, one which is a good place to start. Back in 1933, so we're 80 odd years ago now. And following a basic idea of Galton, they recruited what by today's standards would seem to be a pathetically small sample. But when I was a graduate, actually 100 pairs of twins or whatever was actually quite a lot, you know. Now, I mean, we talk about samples of 5,000. And the reason we talk about large samples is because the bigger the sample, the more precise the estimates you get of the things you're interested in. And actually, one of the very first things I did as a kid, as a graduate student, was not to look at data, but to, I think following a suggestion of my advisor, John Jinks, was to say, okay, if you want to ask the question, is something genetic, for example, a simple question like that, how many pairs of twins do you need to convince yourself that the answer is yes rather than no? If I want to know whether, in addition to the genes, the shared environment plays a role, how many pairs of twins do you need to convince yourself that the answer is yes rather than no? And, you know, it became very clear that, you know, okay, if you want to ask questions, is it genetic? Maybe 100 is good enough. But if you want to start answering more fancy questions, you're talking about hundreds of pairs. You know, and then thousands of pairs. If you want to ask whether the same genes affect males and females, maybe you need thousands of pairs. If you want to look at the interaction, you know, do the same genes operate at different times in development, you need thousands of pairs. And so, you know, this looks pathetic by today's standards, but at the time it was a major accomplishment. When I was a graduate student, this was probably about what we were stuck with. By the time I stopped being a graduate student, we were talking about 1,000 pairs. You know, and so that, you know, now I said, you know, we're talking tens of thousands, thousands of thousands, well, you know, numbers of thousands. Anyway. So, early study. The measurement here there is IQ, and what they did here was to look at just the size of the differences within pairs. So you take each pair of twins, you measure their IQ, and you ask, how different are they? And what you have here is the table of the size of the differences in IQ between twin pairs. Now, all twin pairs, you know, every twin pair grows up in its own home, right? So this was really an attempt to say, okay, let's not worry about the influences of the family background by focusing just on the twin differences. Let's ask, how different are the, within the pairs, are the pairs of twins? And what does that tell us? Well, let's look at the numbers. Monozygotic twins, 65 pairs, average difference 9.2 IQ points. Now, you know, that's a difference. Yeah, you probably find it hard to spot, actually. You know, I mean, I find it very hard if I talk in this room to kind of decide, you know, whether X has got 9.5 more IQ points than Y. You know, really, it's there, but you know, 9.2. So, monozygotic twins. Now, what makes monozygotic twins different from each other within the pair? Take a pair of monozygotic twins. If they differ from each other, what's going to cause those differences? Yeah. What sort of environment?
Unique.
The unique environment. Yeah. The environmental differences that even though these guys live in the same families, they're exposed to different events. You know, I mean, you know, I always say, you know, well, I mean, I drive to work this morning. I have to follow one of those stupid buses to get here. You know, if I'm running a bit late, I get a bit worried. You know, well, you know, I mean, it's that kind of stuff. You know, environmental, and of course, and also long-term things. You know, I mean, you know, identical twins are packed into the same womb, but they pop out at different times. They have different intrauterine environments, even though they share the same mother. You know, they may not have the same benefits of the same nutrition. Maybe in some way competition to each other. Who knows? You know. So there are both long-term environmental influences and short-term environmental. I mean, you know, even the same person on 2 different days doesn't give the same answer. Right? So the slings and arrows of outrageous fortune, the environmental fortune. Okay. Now, dizygotic twins, like-sex dizygotic twins, a good comparison with monozygotes. Monozygotic twins, of course, have got to be like-sex, so let's look only at like-sex dizygotic twins. Mean IQ difference, 17.7 points. Now, why are non-identical twins different within a pair?
They don't have the exact same genetic makeup.
Right, they share some of their genes in common, because they've sampled from the same parents, right? But they've got their own random sample of the genes the parents have cornered. Genetic Mendelian segregation. Right. So, they don't share the same genes. They do, they have different genes, unlike, but what else though? What about, you know, is it just genes?
Male versus female?
Not these guys, because we talk about within pairs, those same-sex pairs.
Unique and
Hold that thought. Yeah. They also have their own unique environmental experiences, don't they? So, in a sense, what you've got here, dizygotic twins, if all things are, other things are equal, also have different unique environmental experiences, both short-term and long-term, that would make them different. They also have different genes. Now, are the dizygotic twins in this relatively small sample significantly less alike than monozygotic twins? Look at the numbers. You're nodding your head. Those who think that dizygotic twins are significantly less alike based on these data, hold up their hand. Those who say, nah, they really, you know, they look different, but actually they're no more different than monozygotic twins, hold up your hand. Don't be afraid. I'm not taking grades, for God's sake. I'm just interested in a conversation. Those who aren't sure. Okay, fair dues. Now, those of you who think that dizygotic twins are less alike, significantly less alike than monozygotic twins, why do you say that? Sir?
If you think of what you mentioned, the standard deviation away from the mean being 15 points, these people were almost 9 points different, which is more than half of a standard deviation away. I think that would be significant.
Yeah. Now, you're going the right direction, okay. But remember, when you talk about the 15 points, you're talking about differences between individuals, okay. we're talking about the average differences within pairs. So we haven't just got 90 or 130 random individuals here, which but we're actually looking at the differences within 65 pairs, and we're looking at the average difference within 65 pairs. So you're right to think standard deviation, good, but we have to go one more step. Because we're averaging numbers that are subject to error, the average standard deviation, the standard deviation of the average is actually a whole lot less. And you can actually do the mathematics, but if you look at the table, actually, 9.2 plus or minus 1.0, that 1.0 is the standard deviation of that mean IQ difference. So, basically we're saying that, okay, 9.2 is the average, but, if you plotted those out, if you did that lots and lots of times, you'd get a standard deviation of about 1 for those average differences.
So each of the people that you study is only like 1 point away from each other.
Yeah, I mean, the within pair differences, right. Yeah, good. Now, so what we're saying here is, okay, so an average of 9.2 points of difference, plus or minus 1. Now, that means if you think of plotting out the normal distribution of average differences, you are going to get differences, it might be 10, probably about 15% of the time. It might be 12 or 11, let's say, 2.5% of the time, on average. Now, think of that, and then you look at so if you say, well, now, how often would pairs of identical twins with a mean of 9.2 plus or minus 1 have the same average difference, if you did the thing again, as the dizygotic twins, who've got a mean difference of 18? The dizygotic twin difference is 8 standard deviations out. If those were, then from what you get with monozygotic twins. So you say, okay, how often, if these were monozygotic twins, how often would I get a difference of 17 if when I know they're monozygotic difference is only 9 plus or minus 1? It's pathetically rare. So you say, well, okay, either this sample is weird, these dizygotic twins are just like MZ twins, really, but, a rare event has happened. Or, frankly, they really are different. So that's why the standard errors are really really important, because they give you a feel for whether the thing really matters. So, those of you who said, yes, they really are, the dizygotic twins really are more different, are right. Okay? You need to focus your reading just a bit more, but you're also right in pointing that the reason you know is because by looking at the standard deviations and stuff. Now, in a sense, you can make this much more fancy. In fact, one of the things that Jinks and Fulker did was to begin to say, well, how can we use the knowledge of how much these samples vary, on repeated studies? How can we use those information to give us an estimate of the precision of our answers? Here we'd say, we really do kind of believe that there really is a genetic component here. Right? Now, you, I think it was, raised the issue of sex differences, right? You said, well, some non-identical twins may be male and female, right? So, we do actually, the genius of Herman and Hogben was they actually included pairs of unlike sex twins, 138 pairs. Now, those of you who decline to answer the question about whether like-sex twins are less alike than like-sex dizygotics are less alike than monozygotic twins. Now, have a shot at the following question. Do you think unlike-sex twins are less alike than like-sex dizygotic twins? Really?
Oh, wait, do I believe in that?
Yeah.
Oh, yeah, I agree.
You think do the data support that position?
Very slightly, though.
Okay. Because you got 17.9 compared with 17.7.
Yeah.
Do the study again. Do you think you get the same answer?
No,
Okay, why not?
it's so close.
Yeah, and your measure of so close is, I mean, you're right, it is so close, it's
Yeah.
like 0.2, right? Yeah. Now, if you don't get the same answer twice, it flips around, like, you know, a little bit. If next time it was the other way around, 17.9 for like sex and 17.7 for unlike sex, you wouldn't be surprised, would you?
No,
Why not?
because they are actually
Go
the
on.
same genetically.
Right. So actually, your answer is quite correct. That actually, although they look a little bit different in
Yeah.
this study, in fact, that difference is not something to take up to write a paper on. You know, this is not a candidate for J-crap stud. You know, in terms of showing there are differences, sex plays a role with impaired differences. Yeah. And of course, the acid test is we can actually look at the standard deviation of the differences, 1.2, 1.5, 1.2. You know, that's a lot of bouncing around. I mean, next, if it's 17.7 this time, it could be 18 or 19 next time. So they can flip around, right? So when you look at the numbers together with the standard errors, you tend to think, yeah, no, there's no difference. The like sex and unlike sex pairs show the same degree of similarity, really. Now, of course, if you do 1,000 pairs, you might begin to find that those differences actually have much more, you know, a little bit, actually are statistically, even though a little bit different. So the problem with doing very big studies, of course, yeah, you get significant results. The trouble is, very often small things come out now to be significant. I mean, do you really care if the difference is small, even though it's statistically significant? Do you really care? You know, does it kind of change your life? I mean, I think the difference between 9.2 and 17.7 might change your life. The difference between 17.7 and 17.9 probably doesn't, right? Yeah. So you have to sort of look at that. Okay, now, siblings. What's the difference between dizygotic twins and siblings?
Nothing.
I mean, biologically, I say different, so I don't mean numerically now, let's just talk about, you know, what is it, you know, what is it about siblings that's not true of dizygotic twins, or dizygotic twins are not true of siblings?
They're not born at the same time.
Yeah, right, yeah. Siblings have, you know, siblings are born at different times in the story of the family. Indeed, unlike dizygotic twins, they're not even packed into the same womb, right? I mean, it's not at the same time. They do come from the same mother, right? Yeah. Well, let's not push that one right now. We'll get back to that another time. Half siblings. We have to maybe half siblings. Okay, so would you expect siblings to be more different than non-identical twins? Yes, no, depends. Yes? No? No? It depends. What's it depend on?
The space and age between each other.
Right, you get it, yeah, okay. So, it's an empirical question, right? If the differences within pairs are caused by the same things that cause differences within pairs of dizygotic twins, then, right, you do expect the average differences to be the same for siblings, right? But, you know, that's not necessarily the case, is it? As you said, you know, I mean, they're parts of different cohorts. They go through history at different times. You know, they had different prenatal environments. You know, probably differences in healthcare. If the mothers have, you know, particularly over here, you know, if the mother hasn't got insurance for one but has for the other, you know, a whole bunch of things can screw things up. Okay, so it's an empirical question. How do we resolve empirical questions? Look at the bloody numbers, right? Yeah. Okay, so let's look at the numbers. Dizygotic twins of unlike sex, well, yeah, okay. Dizygotic twins of unlike sex, 17.9, like sex, 17.7, siblings, 16.8. What's the answer to the empirical question? Or the empirical answer to the scientific question of whether as far as these data go, there's any evidence to support that what makes siblings different is any different from what makes non-identical twins different. No, not really. No, I mean, you know, you've got 16.8 plus or minus 2.3. Next time round, it could have been 17.9 or 17.7, couldn't it? Right? Okay, so, looking at these early data, they illustrate a lot of really important points. I'm sorry to flog this, you know, flogging a dead horse, but you know, really, the answer to any question, if the question can be answered, is you know, it doesn't come out of a computer, it comes out of looking at the numbers and thinking about them. And that's the take home here. So, the take home here, by looking at the numbers and thinking about them and taking into account things like sample size and the inherent unreliability of all statistical data, which is talking about using the standard errors, says really something very simple about the individual differences within families of twins, well, of individuals. The twin data and the sibling data and the like-sex data all point within the resolving power of this study to a very simple initial pattern. Two things are going on here. One is that even identical twins who share the same environment from before birth through most of their, at least, you know, adolescent life, some family environment, are not identical. So even though they have the identical heredity and identical environment, they're not completely identical. On average, they differ by 9 points in IQ, 9 or 10 points in IQ. Non-identical twins, which have, you know, within the assumptions of the twin method, also share the same home, share some genetic similarity, but not all, differ by, you know, actually twice as much. The differences are twice the size. That is consistent with what we know about dizygote twins, is that unlike MZs, they actually do have genetic differences. Now, it doesn't prove it, but at least it's a strong support for your hypothesis that maybe there are genetic differences in IQ, right? But you know, if you've just got those numbers, that's got, you know, you want to be able to say, well, okay, how well does the model fit? Well, you know, we look at the rest of the data. If we look at unlike sex pairs, you know, where sex ought to be an issue, if it is, we find the differences are about the same, at least within this study. So that prediction of the model fits. The prediction is that if it's just genetic and environmental but not sex, you get the same degree of difference. You do. The model fits that. Another test of the model, well, what about siblings? You know, they have different life experiences. They, you know, they're measuring different stages in development, maybe. You know, well, oh, but hey, look, they're no less alike than like-sex or unlike-sex isolated twins. So again, some of the data are suggesting how the world is, and then you're taking that idea of how the world is and saying, well, okay, if it's true, what else would I predict? And that's the essence of the kind of model fitting approach. No, the rest is fancy statistics, but the ideas are really quite simple. Now, of course, you know, in a sense, this is only part of the story that's buried in twins. Because here, quite deliberately, we've just focused on the differences within twin pairs. Because in a sense, it's a convenient way of saying, well, okay, I don't care what makes pair A different from pair B. You know, I've deliberately set out to discount the family background. Right? But of course, that's only part of the story if you think about it. Because pair A does differ from pair B. Why? Well, you know, at least why does this pair of twins differ from that pair of twins? It's considered monozygotics. Would you expect them to be the same? No. Some pairs are going to be really, really smart on average, and some pairs are going to be, you know, average on average, and some pairs are not going to be very smart. So you've got all these between pair differences, right? What causes those between pair differences? Free association time. Don't be bashful. A, you know, I have to preach on Sundays, and I'm very used to the idea that after congregation sits on the back row because they don't want to be looked at, right? People on the back row. What makes pair A different from pair B?
Environment?
Yeah, right. They're different families. They have different mothers, different fathers, live in different parts of the town, right? Yeah, go to different schools. You know, have different teachers. What else?
Indigenous?
Right. Each pair of twins has its own, is sampling its genes from a different pair of parents. Those parents are genetically different. You know, I mean, we're kind of, you know, we might get the luck of the draw, but part of the draw is the parents who have decided to get, you know, to mate and produce us. So, same deal. You know, here we have both genes and environment. The environment now we call it something, we call it the shared family environment, because twin A shares, sorry, the pair A shares a different family environment from pair B, and so on. Or the shared gene, the genetic history of the family, right, is different. So, we have to turn now and say, well, okay, can we use the twins, all the information of the twins, to begin to pick apart a slightly more fuller picture of what's really going on here? Mesdames, Messieurs, let's look at table 5.2. I know, it's almost time to go on, don't worry. We've got Tuesdays, we've got tomorrow's one. Okay. And we'll move faster on, yeah, we're good. Okay, so, look at table 5.2. Don't worry too much about the column labelled mus, okay. Here we have like sex. We have like sex MZ and DZ twins. Now, I'm trying to remember where this data comes from. I think it looks like it's probably the National Merit Twin Study 5.2. Yes, it is. Okay. I think it's the National Merit data. Hang on a sec. Let's just make sure I've got this right. Oh no, this is still the. Oh right, okay, so this is Herman and Hoffman's, this is still the same data. Okay, so now, what David has done here then, now is to go back and pull out Herman and Hoffman's data and say, okay, now let's forget about the within pair differences. Let's just look at the similarity between pairs, on average. And of course the statistic for doing that is what statisticians call the intra-class correlation coefficient. There's a number, look at the column R, okay? 0.84, 0.47. Those are the so-called intra-class correlations for the twin pairs. So I was confused, I was reading the mean squares of sample sizes. They're not, they're the same data as the other one. Okay, so in Word you say the intra-class correlation coefficient for the MZ twins is 0.84. The intra-class correlation for the dizygote twins, like sex, is 0.47. Right? Now, an intra-class correlation is a measure of the average similarity of pairs. So, it's on a scale, forgetting negative values, between 0 and 1. 1, if you've got a correlation of 1, it would mean that each twin in a pair is identical to its co-twin. Different from another pair, but same, perfectly the same. You know, 2 IQs are the same, right? A correlation of 0 would mean that it was like you'd gone out into the street, picked people at random, and then made artificial pairs. That's what you get. You know, no similarity on average. Right. Okay. So, now, one way of thinking about that correlation is to say, well, that's really a measure of the influence of the effects that pairs share. You know, 0.84 is like the percentage, it's 84%. 84% of that variation in IQ that you see in the population is due to effects that identical twins share. a correlation of 0.47 for like-sex non-identical twins is saying, well, the 47% of the variation across all non-identical twins is due to things that the pairs share. Right? Now, based on our previous conversation then, if you've got a correlation of 0.84 for identical twins, you're saying that 84% of the factors that make people as a whole different are the same that pairs of identical twins share and make pair A different from pair B. Now what do identical twins share?
Genetics.
All their genes, right? And their home environment. Right. What David has put in table 5.2 in the last column is model. Okay? And under model, he's put two variables, VG and VCE. And he said, okay, that says, you can write over in that saying, the correlation of 0.84 is an estimate of the joint effects of VG and VCE. G stands for genetic, right? CE for common environment, the environment that's common to each member of a twin pair. So these would be the things like, you know, shared family diet, shared home environment, being in the same school, you know, same socioeconomic status, and all those kinds of things. So, now, if 84% is due to those things, what's the remaining 16% due to?
Unique environment.
Everything else, the things now, yeah, VE, I think probably David would call it, the things known as outrageous fortune. Right, now, diazoic twins, like-sex diazoic twins, correlation of 0.47. That 0.7 is what? Sum of the genes, right? And shared environment. Sum VG and the shared environment. Now, the question is how much VG? David has put the value of a half there, you know, because everybody said, non-identical twins share half their genes in common. That's a very loosey-goosey way of speaking. Firstly, that half is a measure of similarity between the average similarity that you expect between the dose of alleles that twins get. So it's a method, it can be derived mathematically, it's not just but also, to put it there like that makes some very important assumptions. It says, okay, all the genes are adding up in their effects. If you can measure the effect of every gene, they all add up to produce the phenotype. They don't interact in any way. There's no dominance, no epistasia. The second thing is that it does assume that parents mate at random. So there are two assumptions in here which we're making at this point for the sake of argument. And we will look at the numbers and extract the numbers given the sake of argument, and later on we'll say, well, okay, does it really fit? You know, what happens if, you know, do parents really mate at random, and does it make any difference? You know, do the genes add up, or does it make any difference? Now, so, what we have now is, going back to your high school algebra, a pair of what in England we would call simultaneous equations. As I understand over here, you call them simultaneous equations, but I think the meaning is the same. You've got two things you measured, right? 0.84 and 0.47. You've got two things, you've got equations which say these can be predicted by two things I don't know. VG and VCE. But the coefficients of these are different in the two equations. I can solve, you know, remember the old, you know, what is it, 100 equals x plus y, you know, 20 equals x minus y, solve for x and y. Yeah, same deal here. Simultaneous equations. Solve the simultaneous equations. Well, very graciously, David actually does that for us and says, okay, so what's VG? He says, well, look, if I take 0.47 if I take 0.47 from 0.84, right, I'll get whatever it is, 3.7, is that right? 0.4, yeah, okay. So the difference between 0.84 and 0.47 is 0.37. That's on the left-hand side. Subtract the left, stack the top, the bottom and the top, right? If I go to the right-hand side now and subtract the bottom right-hand side from the top right-hand side, the VCEs go out, and I've got a half VG equals 0.37. Now, if a half VG is 0.37, what is VG? 0.37 times 0.2, right? 0.74. So, my estimate of VG based on these simultaneous equations is 0.74. Now, if VG is 0.74, then what is VCE? 0.1, yeah, because I'm going to take 0.84, subtract 0.74, and I get 0.1. So, we have our first overall picture of the genetics of intelligence based on these very early data. Genes, if you believe the numbers and you believe the model, explain 74% of the individual differences in IQ. The shared environment, by contrast, explains 10% of the variation in IQ. And the remaining, that together is 84%, the remaining 6% is the slings and arrows of outrageous fortune. Now, you don't require an IQ of 200 to figure that out, right? The question is, where do you go from here? Well, we now have our first model and our first estimates. The question now is, what do they predict? Do they predict anything? Does the model fit? How might the next generation of science want to modify that? And that's where we're going to go on Tuesday morning. Thank you for your patience, and now read on.